You know, for literally thousands of years, we've been tinkering with the plants around us. It started with the very first farmers picking the best seeds, and it evolved into modern crop breeding. But in just the last few decades, our ability to edit plant life has taken this massive leap forward. We've gone from the slow, patient work of selective breeding to precise, surgical genetic engineering. So today, let's pop the hood and see exactly how scientists hacked nature's own toolkit to build these incredible tools.
So let's get right to it. How does this even work? I mean, if you want to give a plant a brand new trait, say making it resistant to a disease or able to grow in really salty soil, how do you actually get that new piece of code into every single one of its cells?
Well, here's the amazing part. Scientists didn't have to invent this whole process from scratch. Nope. They looked to the natural world and found a system that had already been doing this for millions of years. All they had to do was figure out how to hijack it, how to take the keys and drive it themselves.
Okay, so meet our tiny natural engineer. It's a soil bacterium called Agrobacterium tumefaciens. And this little guy is an absolute master of genetic manipulation. Seriously, it has this natural ability to inject a piece of its own DNA right into a plant's genome. What for? It forces the plant to grow a tumor that then becomes a food source for the bacterium. It's a brilliant, if, slightly sinister survival strategy.
The tool that Agrobacterium uses for this is called the T plasmid. Now, the easiest way to think about this is like a tiny biological delivery truck. The tDNA, that's the cargo it's delivering. And the rest of the plasmid, that's the truck itself, complete with the engine and all the instructions it needs to find a plant cell and inject that cargo.
But here's the thing. This natural delivery truck, as amazing as it was, had some really big problems for what scientists wanted to do. First off, its default cargo was a bunch of genes that cause tumors, not exactly something you want in your corn crop. On top of that, the truck itself was massive, really hard to handle, and its doors were basically welded shut. There was no easy way to swap out the bad cargo for something useful.
So the very first challenge for scientists back in the early 80s was figuring out how to re-engineer this natural delivery system. And their first solution was incredibly clever, but as we're about to see, it was also pretty darn cumbersome. It was called the co-integration strategy.
Okay, so before they could do anything else, they had to make the delivery truck safe. Job one. They did this by literally cutting out the oncogenes, those tumor-causing genes, from the tDNA cargo bay. This created a disarmed plasmid that could still deliver DNA, but it wouldn't make the plant sick anymore. Phew.
But the real trick was getting a new gene in there. Since that big TI plasmid was just too difficult to work with directly, they came up with a workaround. First, they put the new gene they wanted into a small, super simple plasmid inside E. coli. Then they moved that small plasmid into the agrobacterium that already had the big, disarmed TI plasmid. Through a natural process called recombination, the little plasmid basically merged with the big one. It's kind of like bolting a new, easy-to-load container onto the side of the original truck.
And this is what you get. It looks a little bit like a Frankenstein's monster of a plasmid, right? You had these two separate parts that had been awkwardly fused together into one giant thing. And hey, it worked. But you can just imagine how clunky and inefficient it was to have to build this massive hybrid plasmid every single time you wanted to do an experiment.
But science rarely stops at good enough. That clunky system was not the final answer. Pretty soon, scientists had a major breakthrough that completely changed the game. And here it is. This was the game-changing insight. Scientists figured out that the machinery that does all the work, the cutting and moving, called the Ver genes, didn't actually have to be physically attached to the cargo it was moving, the tDNA. Think about it. The engine of the truck didn't need to be part of the same physical object as the cargo container. As long as they were both inside the same agrobacterium cell, the engine could find the cargo and get the job done.
This separation, this was everything. The old co-integrated system was like a semi-truck, where the engine and the trailer were permanently welded together into one giant, inflexible unit. The new binary system? It was modular. It was like having a separate truck and a separate trailer. And this made everything so much easier to design, build, and use.
And here's what that beautiful modular system looks like in practice. On the left, you've got a small plasmid that's just our cargo container. It's small, it's simple, it's easy to load up with our gene of interest. Then on the right, you have the helper plasmid. This is just the engine. It has all the transfer machinery, but no cargo of its own. You put both of these into one agrobacterium cell, and it's like having a driver show up with an engine ready to hitch onto any trailer you've preloaded. The system was now modular and way more efficient.
So this binary vector system, it wasn't just a little improvement, it was a total revolution. It became the new industry standard, the foundation on which basically all modern plant genetic engineering tools are built. So think of these modern binary vectors as a fleet of highly specialized, high-tech delivery trucks. Each one is equipped with what are basically GPS coordinates, those are the tDNA borders, that tell the machinery exactly what piece of DNA to cut out and deliver. They have tracking devices, we call them selectable markers, so we can see which deliveries were successful. And they have universal trailer hitches, these are multiple cloning sites, so we can attach any cargo, any gene we want, really easily. Plus, they're designed to be easily copied in our factory, which is E. coli, before they get sent out for delivery with Agrobacterium.
And these things have become incredibly compact. Those original TI plasmids were hundreds of thousands of base pairs long, but modern systems like the P-Green vector are just unbelievably streamlined. Some are as small as 4.6 kilobases. That's like going from a clunky, gas-guzzling 18-wheeler to a sleek, efficient electric sports car. This massive size reduction made them so much faster and easier to work with, and that just accelerated the pace of research like crazy.
And the impact of this is just huge. It's not just one tool we're talking about anymore, it's a whole toolbox. Today, researchers have access to hundreds of different specialized binary vectors. Each one is designed for a slightly different job, allowing them to create everything from drought-resistant wheat to plants that can actually produce life-saving medicines.
So, from a clunky, reverse-engineered natural system to this elegant, modular toolkit, our ability to edit the DNA of plants has evolved dramatically. We now hold this incredibly powerful pen, one that's capable of rewriting the genetic code that feeds and sustains our entire world. The only real question left is, what do we want to write next?